Where do you play?
Sandpoint Country Club.
Really?
I've been a member there, yeah.
Somewhat like the situation with.
How much did you hear?
Now that there's a
I still
senior group, we play against 4 other clubs in the area. On Wednesdays, 20 of us guys play 20 of their guys.
Well, there's times
At reciprocal, you know, they play us one week and we play them the next week.
Right.
I must have 100 people.
it's going to take me a while.
Tomorrow's our match against Inglewood, who we played at their place last week. It's fun. Play, 18 holes and then have lunch afterwards.
Sounds like a good.
There's a little competition involved. They beat us by 3 points, and we're out to get them this time on our home
Little
course.
competition. There's a lot of competition.
It's nice, the handicap system in golf is sort of equitable.
I was going to ask you.
What's
So,
your
I don't know
handicap?
how much you were joking or not when you said you had me a job, but I
Pretty nice.
How do you set that? You just play a bunch of rounds and then.
Yeah, there's an averaging formula where you enter your rounds and then throw out the worst 2 and the best 2. I don't know that kind of mathematics, but the U.S. Golf Association has this computer system. Where you finish playing around, put in your score, you put the statistics of that course.
Alright. They might have filmed it by now. I know it was open at the quad.
So are you getting better as you play more?
A little.
You just stay the same.
Yeah. A little. Problem is, can't hit it as far. The older you get, you can't hit it as far.
And that way you get the bigger, fatter gloves. If you watch TV, it sounds like all you have to do is get the right glove.
No. That's right.
That's not the case.
Catchy? Cute. Oh, thanks. I feel like I'm dressed for like winter. I feel like I wanted it to be winter. I feel like out there.
No, I went to oh, sorry, I was, let's see.
Morning. Good morning, sir. How are you? Oh, good.
So you play tennis too? Or just golf?
No.
But my ankle way. It's too much.
Baby motorcycle. Baby.
There he is. The only time I see is in national media.
That's right.
And it is everywhere. She's got more bad.
This guy's only having one
Yeah,
film.
I saw that.
Well, that's kind of an austere operation.
I know. Well, that's my. They were looking like a healthy person.
Okay, so I like know.
So you're gonna drop by and pick up the wine?
Friday.
Yeah.
Oh, yeah, and you're bringing some too?
I'm in some wine. Red, right? Red.
Yeah, I got white.
You're
I got
white.
white.
You know where to bring it? You gonna bring
Faculty club, I know that.
Yeah. You know where that is?
And let's park in the back, right?
You would not permit, I don't think permit.
Well, no, for this, for
Drop
the,
off the wine.
I think, you have a sticker, or you in the President's Club? If you got a President's Club, you can park there. You know what, you probably get away with it anyway.
Yeah, you probably get it. So you're gonna bring it to the time of the session?
just before, yeah,
Bring it about an hour early?
yeah.
Okay. Here's what we need. And I'll pick yours up on Friday.
Okay. stays to the airport?
Not really.
Yeah, you know, it's about $200, so I'd rather give you the money.
$200 to go to the airport?
Well, I'm gonna be gone for 2 weeks instead of leaving my car.
Oh, you just take what you call it?
The shuttle for Mars.
No,
Eric can't take? I live in the east side.
No, you hire the
Uber?
Uber or is
We got 5 of us, so.
oh.
In fact, we might have 7, no, 5 of us. What time, you can pick us up,
And then no gun rack.
untwist your arm here.
No options. Yeah. There's a lawsuit. What time is it? Some
Oh, it's in the
Uber
middle of the
driver
morning, like
molested a customer.
10 or 11.
And now the court says they could sue
So you get to sleep
because he was
in.
on the clock when he did it.
Yeah.
We talked to Sarah, but I'm
So I'm sure they were not happy.
They just said the car would be gone for 14 days or something like that. And you know, they charge about 12, 14 bucks.
Uber drivers?
And then we'll worry about coming back. Maybe Jim can pick us up.
That should say something else. I would be afraid of doing that. It always made me a little nervous. I have a bunch of friends who use it all the time. They don't say anything. And you read about some person who get axed. Cut up.
To get your consulting job.
How's it going? Probably not.
Very efficient.
Oh, yeah.
You in the back.
I got always get my head stuff. I hate it.
You know, I'm quitting the
Yeah, exactly.
I didn't write down, you know, easy now.
Last days I had with the whole Amazon state farm insurance for not taking care of their house that got wet and all their
Less than we did. You know, it's like MCS. Yeah, so much less. But, you know, notify them. Stopping at August 31st.
Yeah, exactly.
95.
Get you 32 feet. Never get away with it.
Yeah, conspicuously after.
Buddy.
I like reading ones that I have a terrible time. You know, you rationalize whatever you do.
That's right.
You gotta pick out what you do well. I'm the world's worst eater. This is my kind of stuff.
Pandora.
I get some peanut butter.
Yeah, so he wasn't talking about a little ice.
Yeah, there's a couple real nice areas over there, so you wouldn't want to.
What's that?
I can't remember what it's called. There's, I mean, there's a few.
Steve's in town.
There's some.
I don't know if he's in
Time release.
him or not.
Like he's here every week. Look at this year. Right.
Just that one weekend. You never know. Maybe I'll like it. You know what? I don't even look.
You can walk for $3.
I don't even know. I always have my phone with me.
So it's getting close, is it next week?
Leave Saturday.
No, well,
Anything else?
All right, well, I'll call you. St. Petersburg.
so you were farm
Yeah.
All right, let's go ahead and get started. Everybody take a seat. Just remind you that we only have one more session in this location, and then got the year-end dinner event up at the faculty club on the university campus on the last Tuesday of the year. I think that's the 24th. So I've been sending out announcements about that. I have to go up there today and more or less give them a final head count for how much food we need for that evening. So if you haven't responded and you can come, let me know today. The word of the day is Rochester. Our speaker is Paul Williams. Paul wrote to me about a week ago that while he was researching this subject, he found it now so big he needs to divide this into 2 talks. So he's going to give us half the talk today, and then as we kick off early next year, I think the more clinical part of the presentation. But it actually is an expanding area that's starting to become important in day-to-day practice. So Paul, thanks for coming down.
As Lynn mentioned, this is going to be mostly talking about methodology, limitations, advantages, and so on. And then in the fall, what we're going to do is clinical case scenarios utilizing the tools. And hopefully that'll be interesting to do. So molecular diagnostics, also called molecular analytics, component resolved diagnosis. That term, component resolved diagnosis, is sort of leaving the literature, and now they're talking about mostly molecular analytics. This is our topic for today. I have no disclosures. We're going to talk about limitations of what we use now, skin testing and immunocaps using extract material rather than components, the utility and limitations of molecular diagnostics and or component resolved diagnostics, current state of the art, and some future approaches. Just to run through some definitions that will come up during this discussion. Primary allergen is the original sensitizing molecule. Genuine allergen causes specific sensitization to its corresponding allergen source. You'll hear homolog a lot. That technically is something with a protein with 50 to 70% sequence identity. Actually, over 70% in some cases. If it's over 70%, then often you have cross-reactivity between the 2 proteins. If it's less than 50%, you usually don't have cross-reactivity. Cross-reactivity is just the phenomenon of IgE antibody recognizing or binding to and inducing an immune response to similar allergenic molecules which are present in different species. It often occurs between molecules in closely related species or well-preserved molecules with similar functions that are present in widely different species that belong to the same protein family. So that's what really confuses our process of skin testing nowadays. Polysensitized or polyreactive, meaning positive skin tests for multiple allergens. They may be unrelated IgE responses to several allergens, but also IgE antibodies can bind to several allergen components from a single allergen source. Co-sensitization is genuine sensitization to more than one allergen source when sensitization is not due to cross-reactivity. Principal allergen families we'll talk about a little bit today. We're not going to go into much detail on principal allergen families. That's been the topic of past journal clubs. Major allergen positive means it's positive in greater than 50% of the patients who have a clinical allergy to that source. Now here are the component allergens or inhalants that are of interest to those of us who practice in the Northwest. These are all available either through the ISAC or Immunocap single-plex assays. And so it's just kind of a reference slide. We're going to refer to a lot of these components, and this just gives you an idea about which they are. You can look back at those. These are the foods of plant origin that we'll be talking about. Some of these are limited, very limited in what kind of test you can do. Some of them have multiple components, and you'll see that as we move through. And these are the foods of animal origin.
Paul, is there a system that people name these things?
Yeah, it's basically the original name. For kiwi, for example, it's actinidin, so it's act, and D is and for grass, phleum pratense, so it's the first 3 initials of the first botanical name and the first letter of the second. And then they're numbered based on discovery. 1, 2, 3, 4, whatever. Wheat has about 36 different component proteins. And these are other things: latex, honeybee, wasp, yellow jacket. If you read the literature about venom, you should know that yellow jacket is called the common wasp in Europe, so to mess you up a little bit. And then cross-reactive carbohydrate determinants is what CCD stands for. You could actually measure CCD components using the same company will do immunocaps, will do CCDs if you're curious about those. So what do you need to know when you're trying to figure out what's going on with an allergy and using molecular diagnostics? You really need to know nomenclature and allergen components. And the ultimate cheat sheet, and I'll show you a screenshot of that, is www.allergen.org. All you have to do is plug in the name like dog, and you'll get all the list of all the component allergens that are currently known on there. So it's a pretty handy website. You need to understand some properties of allergen components. So we're going to talk about protein families. It's also important to know about stability, particularly when you're talking about food allergens. Thermal and digestive stability is important. The more thermostable and digestion stable they are, the more likely they are to cause problems. This is just a screenshot, and you can't see it very well, but this is like page 2. If you just enter dog, you can see that this is the result that you get. You'll get the Canis familiaris, so that's where the Can F comes from, Len. And then all the different ones that are studied. There. Okay. So that's what www.allergen.org. That's a good website. So what are the protein families that we're going to be talking about? Well, they're the profilins, which are present in plant-derived food and pollens, serum albumin that are in animals and meat, pathogenous-related proteins, or PR10, that are present in food and pollens, procalcin in pollens, non-specific lipid transfer proteins that are in plant-derived foods and tree pollen, lipocalin, which is in animals, parvalbumin in fish, tropomycin, myosin in shellfish, arachnids, arthropods, and insects, and seed storage proteins, which are present in plant-derived foods. And these are just examples of those different proteins. The important ones in foods are the PR10, the nonspecific lipid transfer proteins, parvalbumin, tropomycin, and the seed storage proteins are the ones that are more clinically relevant for allergy. So BET is birch, BET-B2, FIL-P12, that's a grass allergen, latex is FB8, MAL-D4 is apple, and the little asterisk means that you can get it on the Immunocap Isaac. It's one of the component parts of that test. And the little caret is Immunocap. It's available as Immucap. So you can see that not all of these are available, even though they're felt to be clinically important in some situations. So why do we need to do it another way? We've been practicing, some of us, for a lot of years, just using skin tests and getting along just fine. But the more we study it, the more we understand that there's definitely some limitations in the skin tests that we use. They tend to have variable potencies. Some of them are defined, but they're defined on the basis of major allergen. Things happen to these extracts over time. They degrade, and so you may have missing components that are clinically important. Heterogeneity is a problem. There really are very few standards for quality control for the extracts that we use. Reproducibility can be a problem. They really are a pile of proteins. Both allergic and non-allergic components are present in crude source extracts. As I mentioned, there's poor representation of some allergens, and there's some limited availability, as those of us who've been around for a little while know that we don't have as many things that we can skin test to that we used to, because the FDA in all their wisdom decided that you had to prove safety and efficacy in all the extracts, and they weren't about to spend the money on limited sources like Scotch broom and things like that, and so those are no longer available, sheet, stuff like that we used to have. They also may have some limited sensitivity and specificity. The sensitivity is pretty good, although interestingly venom is not very sensitive. The extract that we skin test to misses quite a few patients with venom allergy. And they all have really poor specificity. We know that positive predictive values of skin tests are pretty low. There are thermo and digestion labile allergens that often just sitting around over time degrade in an allergen extract, so you have less and less positive skin tests with plant food extracts in particular. One of the biggest problems that we'll talk in some depth about is complex pollen exposures and the pan allergens. So patients who are allergic to a large number of pollens may actually be sensitive to a pan allergen that's present in all of the pollens. And so you really don't know what they're truly allergic to if you're just doing skin testing. And the sensitivity and specificity may actually vary with the allergen. For example, venom we talked about, hazelnut is missing some key components in the standard extract. And peanut sensitivity isn't as great as we would think on the skin testing. There's individual variability with the test and also tester variability. And current estimates are that with skin prick testing with crude source extracts, you get about a 6% false negative rate. With serum-specific IgE, you have the same problem. This is the immunocap test, but again, we're talking about with extracts, not component immunocap, but immunocap using extracts, a dog, for example, or cat. Also have limited sensitivity and specificity with a low positive predictive value. They're run about a 16% false negative rate when you do these tests. Interpretation to some extent depends on pre-test probability. We're all familiar with primary care physicians who do panels of immunocaps on their patients and get all these low-level results, and they don't really know how to interpret them because they don't understand the pre-test probability of what's going on. Cross-reactive carbohydrate determinants are really a confusing issue with a lot of different extracts. You have homologs of BET-V1. BET-V1 is the primary birch pollen extract or birch pollen component, but it also is a PR10 protein, and those PR10 proteins are present in a lot of other pollens and plant foods. And so the homologs to that will react positively to BET-V1. And the profilins, that's an example of what we call pan allergens, are present in a wide variety of pollens. And if you're positive to those, then you're going to be positive to all the pollens that you skin test to, but you may not be genuinely allergic to those pollens. Different assays are not comparable. This is an important point. There are currently 3 different companies that have approved immunocap or specific, I shouldn't say immunocap because that's sort of a brand, that has specific IgE testing that are licensed by the FDA. Immunocap is one that's done by Thermo Fisher, and we're all familiar. It's a paper-bound allergen with a fluorescent antibody, anti-IgE. There's Siemens has one called Immunology, and then Hycor is another company that has one. So those are the 3 different ones available. They're all within each other pretty good. They have a coefficient of variability of about 15%, but you can't compare one to the other. You may have as much as 2-fold variation in actual values if you take the same blood sample and run it by one test and then the other. And so it's really important to know the lab that you're using, which one of those they're doing, and make sure that when you're following somebody over time, you always use the same assay when you're checking that.
Paul, when you say that a test has 16% false or misses, what's the gold standard then that tells you 100%?
Well, it depends on the allergen that you're talking about. I mean, obviously with food allergens, it's double-blind, placebo-controlled food challenges. With the pollen allergens or the animal allergens, it's basically just a history. And they've done component resolved diagnostic testing and get better identification of the patients. You have a patient with a history that sure sounds like they're allergic to something, but your skin tests are negative. It's just based on that. So it's not a very exact science, to say the least. And then, some of the results that you get on immunocaps will say less than 0.35, Some of them will say less than 0.10. All of them can go down to 0.10, and the people that are writing these editorials and opinions think that they all should go down to 0.10, but the significance of values between 0.10 and 0.35 is unknown. And it may be significant in some, and you're starting to see probably in some of your results that they have different classes for peanut, for example, than they will for other foods. So they'll say a positive is greater than 0.10 for peanut, but at 0.35 or something else. But some of the people that Atkinson and Hamilton in particular state that really you shouldn't use classes or grades. Those are all totally arbitrary, not scientifically defined by any means, and you should just go with the numbers. But we don't know what the clinical significance of those low levels, and that still needs to be determined. This is kind of a table from Jackie in Practice. They had several articles on this one of last year's issues. It just kind of gives you an idea about sensitivity and specificity of specific IgE antibody testing for different types, and it includes puncture skin test and intradermal skin test. And you can see that specific IgE and puncture skin tests for venoms and drugs are low, whereas ID testing is high sensitivity, but the specificity is really variable. And you get a lot of false positive foods, for example. Specificity is pretty low, no matter how you're testing. And so it just kind of gives you an idea.
Whether to do intradermal tests at all has always been a controversial subject in this room. Some of us do it, some of us don't. You want to comment on that?
That's a whole nother topic.
Yeah.
You know, I personally do intradermal testing for antigens that are lower potency. So I'll do mite and dog and cat, and over time find that you can have a negative prick test and a pretty significant ID test, over 10 millimeter wheel on ID. Somebody made a good point at an annual meeting when Hamilton was talking about this, that most of the studies that look at ID lump anything with a greater than 5 millimeter wheel into the same category. And that may be part of the problem with why some of the people don't feel they have much clinical relevance, because they're taking these borderline tests in a lot of these studies, whereas some people feel that if it's a larger test, it may be more valuable.
So do they comment on the serum IgE? If you have someone with a very, very high level of total IgE, how much non-specific?
Well, we'll talk a little bit about that in later on when we talk about other ways of interpreting these. For certain types of allergens, some people think that a ratio of specific to total IgE is more important, but that's not for all of them, it's only for some of them. So we'll talk about that a little bit. So you can see when they talk about specificity, there isn't a lot of difference between puncture skin test and intradermal skin test, although you tend to have more false positives with IDs and inhalants than you do with the puncture test. So historical factors that influence the pretest likelihood of an IgE-mediated allergic disease, and this helps you interpret, of course, a serum-specific IgE, but we all know this is part of what we do. But obviously, opportunity for sensitization, are they in the right, you know, if you get somebody in Western Washington with a positive specific IgE to ragweed, you know, it probably isn't clinically relevant. Atopic factors, symptom characteristics, biologic or molecular cross-reactivity, specificity of the triggers, and comorbidities, all of which add to the interpretation of these testing. So what can we do these days? Well, we can do a single-plex immunoassay, and that means that's the immunocap, and that means that you can measure specific IgE to a specific component. It's a very quantitative test. You have to have a separate serum sample for each allergen. So if you want, I mean, they can obviously do panels with one draw, but you still need a separate serum sample to run each one of the tests. It's more sensitive at low levels of specific IgE than the immunocap Isaac. And you can get them either with natural or recombinant allergens. And of course, natural are purified allergens, but they have to be careful to try and get rid of the cross-contaminating factors and the non-allergenic proteins that are present in the crude allergen extract. And recombinant allergens, they really have to match the shape and all the details of the natural allergen. So you still need crude source extracts to develop these natural or recombinant allergens. And the multiplex assay, the only one that's currently available is the ImmunoCap Isaac. And it tests 112 allergens. You don't get any choice. If you order the Isaac, you get 112 allergens. That's part of the problem. There's little flexibility in allergen selection, and you may get some results that you don't really want to know about. It is, though, you can do it with a single blood sample. It's semi-quantitative. It's not as quantitative as a singleplex assay, but it correlates pretty well with specific IgE. Results are reported as 0.3 to 100 ISUE. And they figure the threshold for cost savings is about 10 to 12 allergens. If you want to order more than 10 to 12 allergens with a singleplex, you're probably better off just to order the ImmunoCap Isaac. So one of the, I don't know if we'll get into it in the next slide or not, is that one of the problems with the Isaac is it's not very accurate at low levels of specific IgE. And so if it's from 0.3 to 1 ISU, it may not report out as positive when in reality it would be positive if you measured it with the singleplex assay. So if patients have low levels of specific IgE, this test will miss quite a few. So we mentioned that they both use purified natural allergens or recombinant allergens. I wanted to make a slide of the list of the Isaac, but you'd never be able to read it. It's an 8.5, 11 page, very tiny print. I need a magnifying glass just to look at it in paper. But some of them, they're marked with N's or R's. That just means natural versus recombinant allergen. It's really important when they develop the purified allergens from natural sources, they have to establish a degree of purity, ideally more than 95%, determine the extent of contamination with other allergens, and there are very few international standards right now for purified allergens. And so there's no guarantee that one company's purified allergen is necessarily as good as another company's. Recombinant allergens, the advantage are that there's no contamination. They can be engineered to leave out certain things like carbohydrate determinants and things like that. So what are these tests valuable for as opposed to skin testing? Well, you can identify and correct deficiencies in extracts. So that's obviously a research methodology. And some extracts, particularly hazelnut, are now spiked. So they take a natural or recombinant purified allergen and add it to the extract mix because the extract mix is deficient in that particular allergen. You can use it for risk prediction, and we'll talk a little bit about that. You can certainly identify cross-reactive molecules. You can identify genuine allergens as opposed to cross-reactive. Can be used to facilitate patient selection, therapy, and monitoring for allergen immunotherapy, although that's a little bit complicated because of the amount of tests that you'd have to run to really perfectly formulate or select. And it may be the singleplex immunoassay may actually be more sensitive in some situations, and we'll talk about that. Here's an example. This was a Europreval study, so it's done in about 11 different countries in Europe, and mostly for epidemiologic issues related to allergy. And they did one study in peanut allergy, patients with late onset, meaning over 14 years of age, and most of those were oral allergy syndrome, but some of those are anaphylactic reactions. The skin testing with the crude peanut extract was positive in only 20%, but the component result diagnostic test in this group was positive in 60%, so definitely more sensitive for late onset peanut allergy. And in patients who had anaphylaxis late onset again, the extract was positive 70% of the time, but the component result diagnostics were positive 90% of the time. And the same Europreval, they did 731 patients with hazelnut allergy, and again, that did include a lot of patients with oral allergy syndrome, and about 77.4% were positive on prick testing, 83.7% on ImmunoCap, and almost 87% with component resolved diagnosis. So with peanut and hazelnuts, definitely you get better results. The component resolved diagnoses are more sensitive.
Paul, what percentage of those were birch allergic? Did they talk about that? Or?
Well, you know, that's an interesting point that we'll get to in a later thing. The birch allergy and the hazelnut allergy varies by age and geography in Europe. And so if it's Southern Europe, very few of them are birch allergic, and it's Northern Europe, a lot of them are birch allergic. And the kind of allergy they present with in different parts of Europe with the hazelnut varies. So it tends to be more of a severe problem in Southern Europe where there aren't any birch,
Right.
and less, it's more oral allergy syndrome. And it depends on kids. The kids tend to have severe allergy, the adults tend to have oral allergy syndrome. So one of the issues that we'll talk about. So this just is another way of looking at the same four things that we talked about are useful in using these tests. This is the first example, missing or low abundance allergens that are present in allergen extracts. And this allows for the identification of these allergens that are missing or low abundance in an extract. You can either add the missing allergen to the extract, so-called spiking the extract, or you can measure it directly in a separate serum IgE assay if you know what's missing. But you have to interpret with care. For example, hazelnut, if you add this core A 1.0401, you'll get a lot of that's a BET-V1 homolog, so you get a lot of cross-reactivity with people that are birch allergic. But that is deficient and important in some geographic groups. You wonder why that's core A 1.0401? It's because what they've discovered now as they've developed this is that there are different isoforms of these allergens in different epitopes, and some people react more to different isoforms than others. This happens to be a specific isoform of core A1. Wheat, there are a lot of things missing in wheat, and they may be very deficient, very important things that you're missing. So triene-19, which is omega-gliadin, which is important in exercise-induced anaphylaxis, food-dependent exercise-induced anaphylaxis, and wheat food allergy, and you can see some others as well. Soy is deficient, soy extract in gliam-4, and gliam-4 is important. It's a cross-reactive allergen with BET-V1, but in some patients it's significant for severe soy allergy. Peanut ARA-H8 is deficient 10 and 11, which we can't even measure with singleplex right now. Alpha-gal and mammalian meat, we're familiar with that story. Hymenoptera, there's actually only a few allergens that are present in substantial amounts in venom extracts, and they're really variable and they degrade with time, so the sensitivity is low for skin testing the venom extracts. But DESV5, which is a yellow jacket hornet component, is the one that's low. Dust mite DERP P23, it's not very important, but it's positive in about 3% of patients with dust mite allergy who are negative to DERP P1 and 2. So your skin test may miss those. And probably important if you're thinking about immunotherapy, and we'll talk about that later, is that the extracts for therapy are based on major allergen content. So if you have a patient who's allergic to a minor allergen, they're probably not going to benefit from immunotherapy because it doesn't have much of what they're allergic to in it. Latex is missing, has B5. Now, the second aspect of molecular diagnostics that are helpful is you can kind of determine the risk of severe reactions in some patients. And examples are in the cat, FeLV1 is associated with asthma, high levels of specific IgE to FeLV1 associated with asthma. In Sweden, if you're allergic to greater than 3 animal allergens, and we listed 4 opportunities, mouse, horse, cat, and dog, is associated with severe asthma in Swedish children. Dog, interestingly, CAN-F5, which is the prostate-derived kallikrein of dog, is in very low quantity in standard extracts because standard extracts are made of hair and dander, and this is a prostate-derived allergen in dog, and about 38% of patients are sensitive to this allergen, and their history is they only react to male dogs. And you'll miss this with the standard extracts, it would be negative. Cockroach, the per A2, that's the American cockroach. Most of what we test through is the German cockroach or the G2, and that's associated with, the higher levels are associated with severity of lower airway disease. Interestingly, that cockroach extracts are very non-sensitive. They're missing there can be a 7-fold variation in the amount.
Maybe it's covered by the corner.
Paul, while you're finding the slide, you said earlier on, why do we need this? It sounds like we've been getting by just fine. You're pointing out an amazing amount of stuff that we really didn't know we were missing.
Right.
We haven't been getting by fine at all.
Yeah, that's the thing. We haven't been. All right. So with foods, the previous slide was animals. This is a prediction of risk in foods. So we know that in peanut, high levels of ARA-H2 are predicting anaphylaxis and persistence. ARA-H9 is more important in people from Southern Europe and the Mediterranean countries. It's actually related to a peach sensitivity, and so those patients may be actually negative to ARA-1, 2, and 3, but positive to ARA-H9. And in our world these days, where we have people coming from all over the world and living in our area, you have to know some of this stuff, basically. Hazelnut, core A14 and 9, and is particularly important in children for systemic reactions. Core A8 is a non-specific LTP, it's called. It's a Mediterranean folks and related to peach allergen, which is proved B3. I didn't know any of these until I did this talk. Now I can recognize all these. Peach is a real common allergen in Southern Europe, and there's this peach-induced LTP syndrome, which can be associated with severe peanut allergy with ARA-H9. Multiple foods will have profilins, and I put global there because if you're positive one profilin, it doesn't matter what the source of that profilin is. So you can use it just kind of globally. Any positive to a profilin is going to be positive to all the profilins. And so some of the ISAC, for example, some of the profilin sensitivity is low for some of those. So you can just look at the profilins from another allergen source and use that as an indicator that your patient may be allergic to all profilins. And then in multiple foods, you have the BET-B1 homologs, so the birch homologs, ARA-H, the peanut, hazelnut, kiwi, and apple. All of those cross-react with BET-B1, so you can get positive reactions, and those are reduced risk. You get oral allergy syndrome with profilins and BET-B1 homologs. Kiwi, this is one of the, D1 is the primary allergen in kiwi. And it's associated in Iceland with severe reactions, but in other parts of the country or other parts of Europe, it's not. Yeah.
They comment why peaches are important in Europe and not here?
Well, they're important in Southern Europe because of the primary sensitizer. I'm not sure, probably their diet has a lot to do with it. And they don't have the tree pollens that we have that are sort of maybe desensitize them to some extent, that you see with others. So it's always important to know that there's, and this is one of the limitations that we'll talk about later, there are geographical and age differences in these components and what's important and what's not. And attempts to identify cutoff points for either risk for a severe reaction or risk for baked food reactions have generally been not successful. If you look at 3 different studies done in 3 different places with 3 different age groups, you're going to get 3 different cutoff values. And so that's the biggest problem.
Paul, with the reduced risk, the OAS comments, is it felt that it's the density of the antigen per gram? In other words, you'd have to eat so much of it, or is it more binding Did they comment about that?
It's specifically which protein, the component that you're allergic to, is really what they talked about. Although
Gotcha.
they're doing epitopes now, and it probably has to some extent the isoform or epitope that you're reacting.
I mean, it would make sense if it's partly the density, because that's why at standard extracts wouldn't have enough of it, like for peanut, for example.
And then with kiwi, if you're sensitized to ACT 8 or 9, you tend to have more of the pollen-related oral allergy syndrome. So, and apple, MALD3, which is cross-reactive with the peach. In Spain, interestingly, has severe reactions in a third of the patients who are apple allergic, whereas in most of Europe, all you get is oral allergy syndrome. So that's one of the examples of geographic variation. In Europe, in most of Europe, they're sensitized to MALD1, and in Spain, MALD3. And then the profilants we talked about, the reduced severity. So this is kind of a general table for some foods about high versus low risk molecules. So you can see that profilin and CCDs are all low risk. And then there's specific components that they list to be high risk for more severe reactions. And then you can also look at it based on protein families. So high risk are the non-specific lipid transfer proteins, or LTPs, storage proteins, seed storage proteins, the PR10. PR10 is interesting. It can be either lower risk or higher risk, but specifically for soy, it's higher risk, and then wheat is higher risk. Whereas the profilins almost universally are associated with low risk for severe reactions, and PR10s in some situations are low risk. PR10 is the birch cross-reactivity marker. And again, we're not going to get into a lot of details about the specific allergens themselves. Okay, the third use that you can have for molecular allergen is figuring out IgE cross-reactivity. And this is the propeller model. So the shaft of the propeller obviously is common to all three blades. And so profilins and polcalcins are pollen allergen, component allergens that are common to just about everything. So if you're positive to that, you're going to show up positive to birch and hazelnut and oak and maybe even grass and weeds. And the outside of the propeller shows the specific genuine allergen sensitizers. And just another diagram to sort of see these interactions. So they're pollen-pollen interactions with polcalcins we'll talk about a little bit. Plant food to plant food cross-reactivity with the storage proteins and non-specific LTP. And pollen to plant food are the profilins and BET-B1 homologs, and pollen to plant food are latex. Latex are the profilins. So again, just a reference slide. We don't have time to go into a lot of detail, but you can see that profilins are present in pollens and plant foods and latex. And if you're allergic to any of these, you're going to have positive skin tests to probably all of them. And so this is where it helps to use this kind of testing to identify, what are they really allergic to? In the Northwest, we have the advantage of, well, if you're allergic, if your symptoms are all in February and March and you're positive to alder, you're positive to alder. What about other parts of the country or Europe where they have greatly overlapping pollen seasons? It can be really difficult to try and figure out, okay, what's really causing your problem? And so these tests probably have more value in areas where pollen overlaps, pollen season overlaps. The pathogenesis-related proteins, or PR10s, also known as BET-B1 homologs, are present in a lot of pollens and plant foods. And so this is an example from birch, from hazelnut, from apple, and from soy. You can get cross-reactivity of all of those. The polcalcins tend to occur just in pollens, and that's grass, birch, and ragweeds, examples there. And the non-specific LTPs, fruit, and peach is the prime example. In southern Europe, they get sensitized to peach, but then they become sensitized to all other non-specific LTPs. But instead of having mild disease, you can have more severe disease with all of those. So this is possibly the most important use for molecular analytics or diagnosis, with 15 to 30% of pollen allergic patients are sensitive to pan allergens, and so their skin tests positive just about any pollen extract. So you can distinguish between patients with sensitivity to these pan-allergens from those who react to major allergens. It'd be nice if you could skin prick test 2 single allergens, but the FDA is never going to let that happen. And one of the things that people do is they test an irrelevant pollen. For example, they have palm pollen as one of their tests. And if you're positive to palm, you're probably positive to one of the cross-reactive profilins. And so you can pretty much throw out your skin test because it's going to be positive to everything. So that's what some people do. We talked a lot already about the BETV1 homologs. They're common in plant foods. They're usually associated with oral allergy. But the exception is patients who have soy allergies or GlyHEM4 positive. They tend to have more severe reaction to things like soy milk and other soy foods, soy yogurt and things. Profilins, they're positive to cereal grains, but their main sensitization is to grass or birch pollen. And in cannabis, they're starting to see some reports of sensitization to cannabis sativa, and you can be associated with sensitization to other foods that contain non-specific LTPs, such as P3, which is peach or peanut or hazelnut, and even alcoholic beverages, tobacco and latex cross-reactivity and symptoms from those. But fortunately, the corn chips and potato chips are still okay. If you're smoking your weed, you can have the chips.
There are allergenic materials in alcoholic beverages.
You don't care.
Yeah. They're grains and
they carry over into brew things.
They can.
Distill things. Yeah.
okay. And then trying to figure out the genuine or primary sensitization is valuable, again, because of you can filter out the cross-reactivity. These are busy slides, so just want to point out a few things. For grass, for example, and we're not going to go into a lot of detail, but grass had group 1 and group 5 allergens, or FIL P1 and P5. 90% of grass allergic patients react to one of those, so those are the major allergens for grass. 10% may be negative to one or the other, and positive to a few of the other grass. All of those are present on the ISAC, so when you order the ISAC, you get all of those grass pollens. Kind of interesting, we always spend a lot of time, and some patients doing Bermuda grass and other kinds of grass testing, but they did some epidemiologic studies, and there's some cross-reactivity between group 1 Timothy grass allergen and Bermuda grass allergen, which is SYN-D1. And in the epidemiologic studies, they found there are very few patients who are SYN-D1 positive who weren't also FIL P1 positive. So probably don't need to do extra testing to pick up the Bermuda grass patients. You'll probably show up on your standard grass test. Birch, the most important allergen, is BETV1, but unfortunately it's also homolog. It cross-reacts with all the other phagalus trees, which are oak and alder and hazelnut, and so you tend to get positive reaction to all of those. But 95% of birch allergic patients react, and it can be used as a specific marker for birch allergen. But BETV2 and BETV4 are markers for cross-reactivity. Russian olive for people who practice in eastern Washington. Ole E1 is a marker for sensitivity and also for ash trees. Milk, this is a reference for you. These are the different components that are, we know alpha-lactalbumin, beta-lactoglobulin, but it's BOSD4, BOSD5, and so forth. Serum albumin, and 8, 4, and 7 plus lactoferrin, which doesn't have a designation of BOSD something or other, are prognostic factors for persistence, with BOSD5 also being an abundant protein. But there's not been a lot of success in using these to predict who's going to tolerate baked or heated milk, again, because of the geographic and population differences. If you have high levels of any of these, you're unlikely to be able to tolerate heated or baked food. But if you have very undetectable IgE to BOSD5, you usually can tolerate milk, heated milk. And then some of these are also present in beef allergy as well as cow's milk allergy. For egg, we know about ovomucoid, ovalbumin, ovotransferrin, but those are the component names for those. Lysozyme is kind of interesting. You can't measure that, but if you're allergic to lysozyme, you can be allergic to some detergents, and it's It's used as a binder in pharmaceuticals, things like that. Egg yolk, serum albumin, GALD5, and GALD6 are egg yolk, basic. And if you're
How much of this is available on day-to-day practice, and how much of this is research?
Everything that's marked with an asterisk or caret is available. You can order through either the singleplex assay, the specific immunocap, or you can get it on the ISAC. But there are some that are definitely deficient. And then GALD1 and 2 are the major allergens for egg. Cutoff values have not been very helpful. GALD5, the egg yolk allergen, is associated with bird egg syndrome. So people get respiratory symptoms when they eat egg. They get actually sensitized by the bird droppings and feathers. Rat back. Peanut, I think we're probably most familiar with peanut, and people are using peanut component assays a lot. One thing I should mention in response to Len's question is you can order a few component tests from places like Quest. They'll do milk, egg, peanut, but you really can't get very many of these component assays. The only way you can really do it is through Thermo Fisher, unfortunately, and you have to order the specific immunocap for the ones that you want.
And do we know, is insurance covering this?
Mostly not. So that's part of the problem. In the time left, we're kind of, we still have a few minutes. Okay. So I think people are mostly familiar with peanut. ERA 2 and 6 are probably the most clinically relevant. ERA H5 doesn't even come out on the component test, so we don't need to talk much about that. ERA AH is the BET-V1 homolog. That's usually associated with oral allergy syndrome, less persistence. ERA H9 is more associated in Southern Europeans and with peach allergy. And again, some patients may be negative to ERA 1, 2, and 3 and positive to 9 and have a significant risk for reactivity or severity. And that's where he said sensitivity and specificity is population dependent. And interesting, they felt that currently that high levels of specific IgE to crude peanut extract is probably just as good as measuring all these components. You're more likely going to have a severe reaction. So you don't really need to do the component testing very much. They said the same thing about egg. You know, for most of the persistent food allergens, you don't really need these components because it's crude extract. If you get big skin test reactions or high levels of specific IgE, it's probably fine. Honeybee, I think this is valuable to us because of the lack of sensitivity and the problem of cross-reactive carbohydrate determinants with most venoms. The only venom that doesn't have CCDs is the Pollens or wasp. So some people say that it's maybe up to 75% of double positive honeybee and yellow jacket reactions are cross-reactivity and not dual sensitization. And so testing with these recombinant allergens can help identify them. There are a lot of different species of these, and so you can see that there's for example, wasp phospholipase A1, pol D or pol A, or pol A or pol G, depending on the species. But the one that I listed is, and you can see there are actually limited numbers that you can get on immunocap, limited ones that you can get. So you can only get VESV5, pol D5, or APM1 or 4 are the only ones that you can actually measure right now with either immunocap or ISAC. But it's usually enough to distinguish them.
Paul, something you just said sort of contradicts the whole thesis here. You said if you have a big positive skin test of crude extract, don't bother with this. It's, they're highly allergic and you don't need it. Seems contradictory to the entire science here of working this out.
Only for some allergens.
Yeah,
not
and
for all
for
of them.
peanut, for example, I had a patient recently that wanted to go into the military, and his component analysis was positive for 8 and negative for 2, and he passed an oral challenge, even though he had a big positive skin test. It was
Right.
very helpful.
Right.
So it seems contradictory that the people working in this field say don't bother with this if you have a big positive skin test.
Well, they just said it's not any better. Probably not. The component testing isn't necessarily any better in most of the patients who are only allergic to ERA H8 are not going to have a really high specific IgE, like 50 or 100 to the peanut extract. So with venom, the major problem is with CCPs, and so you can use the recombinant allergens to try and find out which one they're really allergic to, so you don't have to put them on 3 different shots if you're going to do immunotherapy. So yellow jacket versus honeybee, you can use VESV5 and 1 and APM1. But APM1 is not as sensitive, so there isn't as much of that in venom extract, so you usually have to use APM4 as well. If you're trying to figure out are they really allergic to both yellow jacket and wasp, you can measure the 2 different group 5 antigens for those. And then just quickly to talk about allergen immunotherapy, some of the studies that they've done in patients who were selected for allergen immunotherapy based on skin testing and crude extracts, they took some of those patients and tested them for per component and found that there may be up to 50% discordance in the selection of allergens for allergen immunotherapy. So I think down the road that may be something that might be useful. We kind of talked about some of them. Patients who have higher reaction to pan-allergens tend to get more local side effects from SLIT. Patients that are high risk for systemic reactions from SCIT, you can kind of predict based on some of these. The more allergens they're allergic to, the more components they're allergic to, the more likely they are to have severe reactions to SCIT. But because of the number of tests that you'd have to do, really tailoring your immunotherapy is not very practical. And I'm going to just skip over this because I want to really get into the limitations, because there are definitely limitations. I mean, when this test came out, you can remember that the people that came out with the ImmunoCap, Isaac, a few years ago, were really pushing this test to allergists a lot. But in reality, the application to diagnosis is really still in its infancy. They really lack some of the clinically important individual molecules, some of whom have not even been identified yet. Some are not commercially produced. Even the ones that they have don't reach 95% diagnostic sensitivity yet. And you still need crude extracts to verify the recombinant components. Native allergens contain many isoforms, so the recombinants have to have the right isoform. It really takes a lot of serum samples from a lot of allergic patients with a wide spectrum of allergen to standardize a lot of these recombinant allergens. There are geographical differences that we kind of talked about with ARA-H9 and CRU-P3 in Mediterranean countries, important in cofactor-dependent anaphylaxis, ARA-H8 and BET-V1 in Northern Europe, so mostly oral allergy syndrome with peanut. You can see similar findings with kiwi and apple allergen. We talked about Spain and Iceland and the differences there. And anaphylaxis to peanuts higher in Northern and Eastern Europe than it is in Southern and Central Europe. So, age differences, we talked a little bit about that in the peanut allergy patients. The late onset tend to be more oral allergy syndrome. Early onset, more severe anaphylaxis. In hazelnut, it's more severe allergy in children, more oral allergy syndrome in adults. The ISAC, we said, was less robust, particularly in low allergen levels. You get a lot of interference by nonspecific IgG4 antibodies because there's only a tiny amount of allergen that's bound to the beads in the ISAC compared to the paper particles or the ceramic particles in the ImmunoCap singleplex. And so low levels of IgG4 can interfere with that, whereas if you have a huge amount of allergen bound to the binding to whatever substrate you're using for binding, you don't get as much interference. There's a lot more of the IgE available. And so you can get bias toward high affinity IgE antibodies. ISAC, the main thing is too much information. You have limited flexibility for allergen selection and low sensitivity to some allergens. What are the unmet needs? What do we not know yet? Well, they really need to do more large-scale population-based multi-center studies to define appropriate patients for this. You can tell from the literature that we've talked about, it's still a little bit of a confusing issue. We know there are a lot of differences. How do we settle all these differences? You need to do large-scale studies on practical use and selection of allergens in the diagnosis. And for that, we need a lot of well-characterized patients and healthy sensitized controls representative of different ages and geographical regions. So you can see where it gets difficult to standardize a lot of these tests and results. The cost utility studies, identification and clinical evaluation of the most relevant allergens, training sessions for clinical and research folks, and development of clinical decision support. So those are what's important still. Some of the other approaches, we talked about total IgE and using the ratio. For persistent food allergens, patients who have high total IgE, the ratio of specific IgE to total IgE may be more helpful. For baked egg tolerance, there's one study that suggests that serum IgE, the specific IgE ratio to IgG4 may be important. You can do a basophil activation test, which is probably more important or helpful in patients who have very low total IgE. In other words, patients who have a negative skin test but a positive history. Some groups are doing what's called the basophil allergen threshold sensitivity test, which is the lowest allergen dose to upregulate CD63 by 50% of the maximum. The future, what's going on now? What other things are people trying to decide? Well, they're multiplexed allergens. So for dog, for example, they're developing a test that has all 4 components in one multiplex. And so you get a much higher sensitivity with those. There's suspension arrays where you can do lower amounts of allergen groups, so you don't have to do 112. They use bead technology, so you can select different beads. Hydrogel biochips, which are a smaller scale than the ISAC. And then right now they're doing epitope mapping with this surface plasma resonance imaging microarray. Can't even pronounce it. These are suspension arrays, so these, number one is a little bead, and they bind the allergen to the bead, and then they have the patient-specific IgE and an anti-IgE and chemiluminescent marker. So they can actually mix these beads. So if you want to do specific groups of allergens, they can just pick the beads out and throw them into the suspension array. So it gives you a lot more flexibility. One of the issues they're really trying to focus on is point of care. So they're trying to develop biosensors where you can actually do it at the bedside and get your results in about 15 minutes. Scary thing that they talk about the ImmunoCap Rapid that's going to be marketed in clinics and pharmacies, but you can get the results in 15 minutes in those, and they use a gold sort of binding system.
That's in development?
Yeah. It's actually trademarked right now, so it's not commercially available. But
so you don't need allergists anymore, you just the pharmacy does the diagnosis?
Yeah, pharmacy, Costco.
Been doing that for a while.
Right next to the Flonase on the counter.
You guys are training.
And we don't have time. So to go into detail about some of the techniques, but epitope mapping is probably where we're going next. The molecular diagnostics measures responses to proteins as a whole, but epitope mapping actually evaluates the IgE binding to specific segments of proteins. You can determine conformational versus linear epitopes, and of course the conformational are less likely to persist compared to the linear epitopes. They're doing newer techniques using peptide microarrays, and they're trying to solve the age and population related differences. So, you know, they're just trying to figure out what's going on with all these epitopes and all these proteins and components, and now they're developing new ways of looking at it. All right, Paul.
So when they're making these tests, are they going back to check the extracts to see if the extracts actually contain these specific subproteins?
Yeah, that's what they've done with some of those spiking of the extract. They've gone back and figured out, well, you know, this patient was allergic to this by history and by challenge, and yet their skin test was negative. What's missing? And that's how they discovered some of these.
So are they going to do that for every one of the extracts, or are they just going to do it for the ones that they've identified so far?
Well, they're in the process of trying to identify that for all of them. And they've just identified, well, hazelnut and dog were the first common one.
In clinical practice, as you know, if you do a skin test and an extract and it's negative, sometimes you'll bring in the fresh food for the skin test, even though that's not standardized. It's been shown to be more helpful.
Well, it is for some foods, but the problem is the specificity is pretty low. You get a lot of false positives with fresh food testing. It's kind of interesting. But sesame, for example, is the extracts are non-specific too. So. I mean, one thing I do learn is that the skin testing that we do is not an accurate science by any means. It's definitely got its limitations.
Chuck Jackson wrote in a question here, does sensitization to pan-allergens like Bet-V1 or Propylene mean that they can trigger symptoms as a component of any of the pollen or food containing them? Or are they a source of false positive tests without being clinically significant?
Well, the answer is yes to both. I mean, mostly with the pollen and food cross-reactivity with profiles, you get oral allergy syndrome with the foods. But if you have a patient who has, if you're measuring components, and they have positive reaction to profiling, you can pretty much throw the skin test results out the window because they may be reacting only to the profilins and all the pollens and not any of the major allergens.
Well,
then you have to
and isn't the idea that if you put all 9 extracts for those pollen, that's where you're going to increase the risk of reaction?
Right.
I mean, because you're inadvertently, it would be like doing the same antigen 9 times.
Well,
Now, granted, the density might be less, but that's still a lot.
well, but the profilins tend to be fairly safe.
Right.
So,
But like LTPs or something.
well, LTPs, yeah, that can be a real problem. But it seems like peach is the
Right.
prime mover for that problem. So
Yeah.
we don't have a lot of that here.
Yeah,
Certainly in
exactly.
Southern Europe.
Each and mugwort.
So what of this is ready for prime time that we should be incorporating in practice?
Come back in
Today.
September.
Yeah.
Come back in
Well,
September.
Stay tuned.
In a lot of ways, we've got an advantage here in the Northwest because we have fairly well-defined pollen seasons. And so you can pretty much tell from the history which of those positive skin tests are reliable. But if you get into areas where there's southern U.S., where there are large overlaps in pollen seasons, it can be really tough. And so these tests can help you identify, particularly with immunotherapy, what you're going to be doing. What's the appropriate one? Venoms, I think, are a real benefit right now in prime time because of the high level of CCGs and venom extracts and the low sensitivity. I think this might be helpful, but no one's really offering much in the way of venom component tests. You pretty much have to order the Isaac or you have to order the individual component tests. And the other issue is that insurance companies aren't paying for it. They still consider it largely investigational.
So we shouldn't write venom orders based on skin tests because we're often duplicating unnecessarily?
I don't want to say we shouldn't because we don't have a lot of choice, but we're over-treating a lot of patients. We're doing too many extracts that they probably don't need. We don't have any way of distinguishing. I mean, if there's a huge difference in the skin test reactivity, you can kind of guess, but that may be because there isn't a lot of venom in the extracts. You get patients with a positive at 0.1 or 0.01 to yellow jacket and only 0.1 or 1 to honeybee. It may not be because they're more sensitive to yellow jacket. It may be because there's less honeybee venom in the extract. There's a less sensitive extract.
So next year we'll come back to
clinical scenarios.
Thank Paul.
There's a literature about local allergic people who have negative skin tests, which blow stuff up their nose and they react. How does that play out with this?
I don't know. I didn't look at that. That would be an interesting topic for journal club. I think is the local allergy. There's more and more stuff coming out outside. Yeah, but
You got a lot of people that tell you, March.
But you still have to have something. So, those patients typically are negative on serum-specific IgE as well as
Exactly. Well, and the third leg on that stool is to do an allergy immunotherapy that could give them some local stuff.
Yeah, well, you don't know what you may not be giving them what you need to give them either. You just guess.
Well, it's in there. In the extract, if you blow it up their nose.